So, welcome back, everyone. What we will focus on now is AI in drug discovery. So it's a very applied area. So I'm Andreas. I'm both here at the university in the chemistry department, actually. So I'm a chemist by training, but then programmed when I was young and so on. So I always wanted to combine data analysis in the life sciences. And I'm interested in both academia and in startup companies as well. So we started now 3 companies in total. So this talk will be a bit different from the other talks, which were very heavy on algorithms, for example. This one here is focused on one domain. Yeah? So how can we use algorithms in the drug discovery area to really solve problems? Yeah? So this is not about publishing a paper or so. Yeah? It's about doing something applied and solving a real problem. Because it's very important to really understand the problem domain. To find out which data is there, what does the data mean, how can I build a model to address a real problem in that particular field. And that's quite often different in a pharmaceutical company, let's say, or in a research group, in a PhD student or so. It's tempting to have a model where you optimize whatever area under the curve or correlation coefficient or whatever your loss function is, right? But in a pharma company, for example, the question is, okay, your model predicts something. So what is the assay? So the test, the experimental test you can do to follow up on that prediction, right? You don't have that question if you just train a model, but you have that question if you work in a pharma company, and the question is what to do with the prediction, right? So sometimes that environment can be really different. And so what I will shed light on here over the next hour or so will actually be how can we use data in drug discovery. And much of that is actually published last year, at least the main thoughts, in those Drug Discovery Today articles. And so I can really recommend them. They're open access, so everyone can look at them. They received quite some coverage, so I was talking to Bill Gates and Angela Merkel as a result as well. And so that actually outlines the problems we have with data in the drug discovery environment. It's very different from the image domain, for example. It's very different from speech data. The data we have is much more conditional. Yeah. So if you generate data in drug discovery, the readout you get depends on the concentration, depends on the time point. How you react to a drug depends on you, your genetic setup, for example, right? And that's very specific to the drug discovery domain. And that's what I will outline over the next hour or so. And I can recommend give those papers a read as well, in case you're interested in the topic. Today will be the first part of the presentation, tomorrow the second part. So we will talk a bit about the current state of AI in drug discovery today, and then there will be a good part of today's talk about data. Yeah, and that is what I would call the Achilles heel of using algorithms in the drug discovery field. Quite often we don't have enough data, okay, but that's like that in many fields, but quite often we have the wrong data as well, and we don't really realize it when we train models. So this is what we will go through today. And tomorrow it will be a bit about methods validation, and also a bit about psychology, right? When I was young, I also thought decisions are made based on science and scientific results, right? But quite often you have psychology that comes into play. There's, for example, certain areas that are much more well-hip, much more perceived as prestigious than others, yeah? And that, of course, drives decisions also in companies. And so we will discuss that very briefly. Translation, so how do we actually translate a machine learning model into a functional drug discovery, into a functional model in a drug discovery environment. And there will be some examples as well on generative models, for example, that we will go through tomorrow. So where are we right now? Sure, yeah, and ask questions, please do. Good question. How is it? Yeah, I'm not quite sure it's actually on me. Is it on?
To switch it off.
What do you need to press?
We need the microphone to record in Zoom.
No, turn up, right? Turn up the microphone.
Louder. I have no idea.
Here there's nothing. It's not even on, right? Oh, no, it's
something main menu.
Audio
menu. And now we just try to put it.
Okay, hello, is it better? Hello, does it work better? Is it okay? Yeah, like this is okay? Yeah, like that's better? Okay, very good. Yeah, it's better. Yeah, okay, so thanks by the way. So where are we right now when it comes to AI and drug discovery? And today we are in what I would call the third wave of using computers in drug discovery. It's always important to put things into context. Yeah, using computers in drug discovery is not new. It's been done for, well, at least 40 or 50 years. So this is the fortune cover from 1981, where the next industrial revolution was proclaimed. And that was a year when Silicon Graphics, for example, was founded. It's one of the companies that really revolutionized graphics visualizations, graphics cards, basically, right? That was 1981, yeah, so it's 40 years ago. That was also the area when, for example, dock, so that's a software where you can use ligands, so chemistry, to dock them into proteins, was published as well. And so this was a fortune cover from 40 years ago where the next industrial revolution was proclaimed. Yeah, and that was 40 years ago, right? And then there was another wave of computers and drug discovery in 2000. So this first wave was very much graphics driven, yeah, visualization driven. Then there was another wave around 2000. Some of you might remember or might have heard of at least of the biotech bubble, right, around 99, 2000. That was the year when the human genome was sequenced as well. And that was much more data driven. Yeah, that's also when cheminformatics became popular, for example. So in the 80s, the first wave, that was graphics. Around 2000, there was a much more data driven wave. And more recently, the headlines that we see, they actually sound pretty similar, right? I mean, the next industrial revolution, and here how AI is changing drug discovery. So it sounds similar. Are we actually in a similar situation? In some aspects, maybe yes. In some aspects, certainly not. So what changed is we have GPUs, we have TPUs, we have much more powerful ontologies. Yeah? It's very important how you represent information, how you structure information. There's now ontologies when it comes to drug discovery information. We didn't have that 40 years ago at all. But what is missing certainly today is the question, which data do we need to answer a question? Yeah, and which biological data, genomics information, proteomics information actually tells us whether a patient will respond to a certain drug. Yeah, and those questions are still unsolved. And these are actually the most important questions because those questions tell us which data we need to generate to answer a question. If you have a drug and you want to bring it to the market, two things matter. One is efficacy, the other one is safety, right? So do I have the right drug to cure my disease? And will the drug actually make it worse or not, right? So efficacy should solve the disease, but not at the expense of incredible safety problems, right? So that's the two only questions that matter in drug discovery. And then the question is, does sequencing data, for example, tell us who responds to a particular drug? And the answer is only to a very small extent, right? So the problem is still which data do we need to actually pick the right compound for the right patient? And that's still unsolved to a great extent. So this is where we are today. So it's kind of the third wave of AI and drug discovery. There was quite a lot of funding going into the area. That estimate here is maybe 20 billion or so so far. And I still remember, so in case many of you probably want to start your own companies at some stage as well. There's one thing I learned from starting our companies. The first one was Helix in 2014. And it fell late to start that company in 2014. If I now look back, it's 8 years later, yeah, AI is only now in full swing, or was a year or two ago. If you think you're late, quite likely you're not late, yeah, because if you're an expert in the field, you're probably on the forefront of science, yeah, and probably the whole field only moves much later. So if you want to start your own company, don't be distracted by the feeling that you're late with what you do. You're very likely one of the first movers. Yeah? So we started Helix here in 2014. When there was hardly any investment going into that area. And so this is the funding that went into the area in both startup companies as well as bigger companies, of course. So what do we get for that, right? Money in is only input, but the question is, what do we actually get for that? And so this is what the current drug discovery pipelines look. And that is on the left-hand side here on this plot. It's for, on the left-hand side here on this plot, it's for, okay, because it doesn't go. Okay, okay, so this one. So here on this plot here, that's for companies that are AI native companies. And so that's companies that put AI computational methods first, basically. And on the right-hand side, that's the plot for big pharma. And there's different phases in drug discovery. First, you're basically in the test tube. That's what's called discovery. Then you have preclinical phases, so you're in animals. And then you have phase 1, 2, 3, when you go into the clinic. And you treat patients, right? And phase 1, you discover safety of compounds, and phase 2 and 3 tell you something about efficacy. Can you cure the disease, right? And what you see is if you look at companies in the AI area, so that's the left-hand side plot, you see that most of the projects are actually green, right? So what does it mean? It's very early stage projects. Yeah? It's compounds that are not yet in humans. Yeah? And so what does that tell us? We actually don't really know whether AI in drug discovery works, right? Most of the projects are still, even 10 years after the bubble started, even now, they are actually in very early phases, right? And so we don't really know whether AI in drug discovery actually is a step change or not. It's too early to say. And that distribution of projects looks very different in bigger pharma companies on the right-hand side. Right, you have a distribution across all of those phases. So you have some projects that are in discovery, but you have many more projects that are actually in the clinical phases, right? So that's much more balanced, yeah. So you're much closer to the market, basically. And so that is actually, and that will cause problems over the next few years, right? So right now, money becomes much more expensive. So what does that mean? It's more difficult for startup companies to get new money, right, for next funding rounds. So either you bring a product to the market or you fail, right? I mean, that's basically what will probably happen to some of those companies. And in case you're interested in the field, there's also this Nature Reviews drug discovery article that I can recommend as well, that actually outlines that in a bit more detail. So which types of targets do companies actually work on? So what is actually a target? So a drug target is a protein in the body, right, that you want to develop a drug for. And so there's, well, 23,000 genes or so you have in the body. So maybe you have 10 different proteins that those genes are mapped to or so. So you have a few 100,000 proteins probably in your body. And then you can develop a small molecule that modulates that protein in order to cure a disease. So there's, for example, a kinase, as it's called. So it's an enzyme that's overexpressed. You have too much of that, and you can develop an inhibitor that inhibits that kinase to cure that cancer, for example, right? And what you see here is the distribution of targets that companies in the AI field, as well as in conventional pharma companies work on. And so these are the targets that AI companies work on, the companies that big pharma companies work on. And what you see is that AI companies actually focus on relatively conventional targets. These are areas where there's lots of data known and where that target pharmacology, as it's called, is relatively simple. So you have some proteins that can do many things, G protein coupled receptors, for example. You have different downstream signaling pathways and so on. And you have other proteins that can do only one thing. You can block them and the problem is solved, okay? And in those cases, the pharmacology is much simpler. And what you see here from that plot is that there's more focus on enzymes, kinases that have simple pharmacology. So they can have a compound that blocks that particular protein and you actually cure the disease in the best case. Yeah? And so this is actually, so AI and drug discovery companies are actually quite conventional in some way. Yeah? They focus on areas where there's lots of data, which is actually the areas that pharma companies worked on for quite a long time. Okay, so this may be a bit of a boring conclusion here. There's little novelty basically in what those companies do. The same is true if you look at the chemistry. So if you have a look at the discontinued drugs, they're in gray here, the ones that are derived by chemists, so these are in red here, as well as the ones derived by the computer here in green, and you see that they fall in similar areas of chemical space. So that means from the previous plot, right, there's focus on relatively explored target classes, proteins, and there's also a focus on relatively similar chemistry. Okay, so that's a UMAP plot, so it's a projection from high dimensional space in 2 dimensions, can be very misleading, but in this particular case, actually, it's very similar chemistry. So it's relatively little new chemistry, basically, that those companies come up with when it comes to drug discovery. Yeah, so both in the target domain as well as the chemical domain is relatively conventional and maybe a bit boring things that are being done right now. Okay, so what can we conclude? So there's lots of activity in the early stage pipeline of AI first companies, but it's quite often already explored targets, close analogs. Data is often the limiting factor. Yeah? You can usually, you can train a model based on data or you model based on first principles. If you do that based on data, yeah, you need to take what's there. And that's often data for chemistry that has been worked on for decades already. And I always think about if companies, if you read press releases, sometimes you have very big funding rounds, in vitro recursion and so on, 400 million or so, yeah? That is only input, right? The question is then what do you deliver with that? What is actually the output? And I think that is still what we are a bit waiting for. And if you have output, the question is it only technical progress or is it in vivo relevant, okay? And that's the concept. If you ever work in drug discovery using computational methods, the question is not how do you get a better AUC or model performance, right? How do you actually bring a compound to the market that is safe and efficacious, right? That is always the question, and that is the term here, in vivo relevance. Yeah? I will come back to that question in a few slides, There's simple data that we can generate very easily, but that is precisely not really in vivo relevant because it's test tube data, so to speak, derived from very simple systems. In vivo relevant means does it actually relate to the effect in the living being, in the human system. The first AI-designed drug will certainly be celebrated by the media, but you always need to make sure to compare what happens to the null model, right? We've seen before that 20 billion or so of funding went into that area, right? It costs $1 billion or something to discover a new drug. Yeah, so what would you expect from the null model? I mean, you would expect 20 drugs or so, right? But of course, that's then a biased perception by media as well, right? If there's the claim that an algorithm has then discovered a new drug or so, you would expect that to happen by chance even, right? That is actually, so you need to see what is your baseline when it comes to model performance. A few slides about AlphaFold, because I'm pretty sure many of you have seen that in the media. So what did AlphaFold do, and what is the likely impact on drug discovery? So it was even reported in the New York Times, right? Of course, in Science and Nature and all those magazines. So what did AlphaFold actually do? So if you have a protein, then you have it in your DNA originally, right? Then it gets expressed, so the genes get expressed, and then translated into a protein. And those proteins, they are in a one-dimensional sequence, but then they fold. So they have a three-dimensional fold as well. And the question is, based on the sequence, what is actually the fold that the eventual protein will assume? And so you have a one-dimensional sequence at first, then you have secondary structures, so beta sheets, alpha helices, and so on. And then they fold further to form a tertiary structure. Okay, and this is what AlphaFold was trying to predict, and actually succeeded in predicting as well. So it was a deep network architecture. I don't go into the details of the architecture here, but how it worked is it did a multiple sequence alignment. So try to learn from tens of thousands of structures where you can compare the sequence and conclude what's actually the fold that sequence will translate into. And at the end, at the output of the model, get a 3D structure with assigned confidence scores as well. So you know where's the model more confident and where is it less confident. So you output that 3D structure and the confidence. It outperformed many other methods. And where does it actually have an impact in drug discovery, right? Because that was, it's one of the questions. You have a method that does something new. How does it translate to the drug discovery field? And if we evaluate that a little bit, so where does it actually help? It helps solve the structure of new proteins. That's an area that takes quite a lot of manual work otherwise. Yeah, you have an electron density from an X-ray structure or so, then you need to empirically linearly fit the sequence into that model. That can take quite a lot of time. That's where AlphaFold can help. You can also design new proteins with desired functions. To an extent, there's quite interesting papers on that. And you have the protein structure, which you can use, for example, for docking, so computational method to fit a ligand, a potential drug, into the binding pocket, into that protein. So this is where AlphaFold would help, for example. But where it doesn't help directly is actually drug discovery when it comes to that in vivo translation. So if you have a structure of a protein, the question is, if I modulate that protein, what is the in vivo effect? What is the effect in the living system, right? And this is where AlphaFold basically stops, right? You stop at a structure of that particular protein, which is a good start, but it doesn't help going into the in vivo direction. What is actually a characteristic for that problem? And that's two things. And these are also hurdles when you think about potential future applications of machine learning and drug discovery. The first one is the work was actually based on decades of experimentally solved protein structures. So that means there were tens of thousands of protein structures that have been solved in the last decades, which could be used in order to train the model. The other one is clearly defined labels. In drug discovery, quite often you're actually not able to label your data sets properly. I will come back to example in a few slides. But here you actually have clean 3D structures, so you know what you actually want to learn. And that, as I will illustrate in a few slides, is actually difficult in many cases in drug discovery. So basically it solves something at the very beginning of the drug discovery process, having a structure, but then the question is how does that translate to effect in the in vivo situation? And that is something that is not addressed by AlphaFold at all. So let's talk about data and drug discovery a little bit. So to explain three words here, and these are very important words. If you ever work in the drug discovery field, that's very important to distinguish. And that's in vitro data, in vivo data, and the translation of one to the other. So drugs work in organisms, right? Usually in humans, but of course you have drugs also for animals and so on. And historically, those drugs were discovered also in organisms. For example, in pigs or in other model organisms. Pigs are quite good when it comes to immune system or lung function, for example. So historically, drugs were discovered in living beings. Then in the 80s, actually a slightly schizophrenic concept basically appeared, and that's high throughput screening. Yeah, and that means you simplify your system, so you don't do drug discovery in whole animals anymore. You isolate one protein and you actually test that in, well, test tubes or those microtiter plates here. And so you test many, many compounds at once in those very simplified systems. So you have many more shots at the goal, you can generate much more data, but it's only a very simple system, right? It's only a proxy system. Yeah? And since then, there are actually two directions in drug discovery. One is the in vivo direction, so you're in a complex system, right? You have all the functions of a human being, for example, in your system, or you have a very simple proxy system, a very simplified system. Yeah? And there was data generated on both levels. Yeah? One is in vitro data, that's the simplified data. The other one is in vivo data, that's data in the living system. And then translation means that we can take measurements from one system, the simple system, and we can translate them to the in vivo system, right? And that's very important. And if that translation doesn't happen, it doesn't matter what I measure in my simple system, I have lots of irrelevant data, right? And that's a problem when it comes to much of the data that we have. So if you think about AI and drug discovery, what do people actually currently look at? And quite often that is actually not drug discovery. It's more compound activity on a single protein. So if you take a drug, what happens to that drug? It dissolves in your stomach, right? It distributes across different organs, it might accumulate in the liver or in the brain, for example, right? And this is what's called pharmacokinetics properties. And then it gets metabolized, gets excreted, and so on. It might have secondary targets as well. So it acts on one protein, but also many others. So you have side effects, for example. And that whole profile, that determines how a compound acts in the living being. What do many people look at if they do AI and drug discovery? They actually only look at a single aspect, and that is the activity on a single target. Yeah. So basically, you have this protein that gets predicted, for example, by AlphaFold, and you only look at the interaction of that compound with that particular protein, but you neglect all the other aspects of compound function. Okay, and so that means it's a much smaller slice, basically, that gets addressed by many of the algorithms right now. And that means there's hardly any real drug discovery around right now that gets addressed by AI algorithms. So where do actually better decisions really make a change in the drug discovery pipeline? So what we simulated is the impact on model success across the drug discovery pipeline. So hit discovery means you find a compound that's active on a target, then you optimize that compound, then you're in preclinical, so animal phases, and then you're in clinical phases, phase 1, 2, 3. And what can change in drug discovery is you can be faster, right? You can have better compounds that go into the clinic, or you do something more cheaply. Yeah? And what you see is that the black bars here in the clinical phases are highest. So that means you have the failure rate that is reduced by, in this case, 20%. Yeah? And so that means we need better compounds that go into the clinic that are actually efficacious and safe. Yeah? But unfortunately, much of the algorithms and the data we have is actually located here. So in the much earlier phases of drug discovery, where the bars are lower, right? So there's less to gain, basically, by using algorithms. And there's two different types of data that we have in drug discovery, right? I alluded to that already, in vitro data and in vivo data. And one is actually simplified view on data that can actually help in practice still. But in vivo data is what we actually want to address. We want to have efficacious and safe drugs, right? And that's two different viewpoints, and which I will go through, and two different types of, well, algorithms that you can use in both areas. So the first one is in vitro data. So that is compounds we can label properly. If you think about supervised models, for example, you need to label your data to build a supervised model. Unfortunately, we can only do that with in vitro data. With in vivo data, it's difficult because our data is so conditional. I'll show that in a few slides. And that's also one of the reasons why algorithms that you have in image and speech recognition, CNNs, RNNs, and so on, don't really translate to drug discovery, and that's a big problem. But there are conceptual problems, basically, why that doesn't really work. So this is a simple view on the world, about myself as well, until 2010 or so. So this is the typical data you would have from the chemical and biological domains if you want to do drug discovery. So you have chemical structures, and you have bioactivity data, There's Campbell, PubChem, public databases around. So if let's say 100 million or so data points or so that link a chemical structure to a few thousand or so protein targets, then you have pathways. So these are annotations that group proteins in particular biological functional context that link a set of a few dozen or so proteins to a particular pathway that regulates growth, for example. And then you have links between chemical structures as well as phenotypes. Those 1,000 compounds cause liver injury, for example. So this is the data you would deal with. And if you can label the data, and in this case we assume we can label our data, we can actually build supervised models. They are quite simple models in many cases. They can be useful, but they're also limited. And those labels could be: a compound is toxic, a compound binds a particular target, or a particular compound treats a particular disease. In many cases, though, that's a very simplified view because all of that depends on dose, for example. As we will see, water, for example, you wouldn't describe it as toxic usually, but water under certain conditions can also be toxic for humans, right? So that shows that labeling data can be very difficult in the drug discovery context. But it works where the data is large scale and homogeneous, and we have meaningful labels, okay? And what can we do in those cases? We can actually, if we go to that triangle here, we can usually take data from two of those domains and we can infer the third. So we can, for example, say, I want to cure a particular type of cancer that is driven by a particular upregulation of a certain gene. And then we can, based on the bioactivity data we have, we can say, give me particular structures that are likely to address that particular protein to cure that cancer. Or we can say, if I have a particular, for example, HIV, that works very well. If I want to decrease the HIV burden in a particular patient, then I know which particular enzymes I need to hit in that HIV patient. So what is the right chemistry, even in a personalized way? What's the right chemistry, the right drug to give to that patient, right? So I can usually infer the third corner, basically, of that triangle using the data I have. And we can do that, for example, for target prediction. So we can say if I have compounds that have a certain effect in the living system, so compounds that are toxic, for example, or compounds that cure particular cancer, any effect in a living system, then based on data, I can predict, for example, the protein target. So what does it look like in practice? So that's a model we built with AstraZeneca, for example, a couple of years ago. It's actually quite a simple setup, but in practice it's difficult because of biases in the data to deal with that. So if you have particular areas, particular ideas how to improve that situation, they're more than welcome. But still, by and large, an unsolved problem. So what you have is you have in total hundreds of millions of data points, but you have what's called active molecules against the target and inactive molecules against the target, right? So you have a class label that's activity against a particular protein target, and you have, let's say, 20 to 1,000 or so active molecules and 1,000 to 10,000, maybe 100,000 sometimes, inactive molecules for that particular protein. And then you have a set of a few thousand proteins you have data for in total. So you have this setup basically a few thousand times. And then you're training a multi-target, multi-label classifier effectively. Depends on how you set up your model system. But you're trying to predict absolute class likelihoods of a new molecule to any of those active classes that you have data for those thousands or so of proteins. And it's a difficult problem because you want to predict absolute class likelihoods in a space where you sample very unevenly and where you have lots of data biases. Yeah? You have lots of analogs, as it's called chemically. So in some cases, you have 20 times basically the same molecule in the data set. In others, you have much more diverse sampling. Yeah? So it's very difficult to make predictions in that space. So you have a few 100 million or so data points. You can encode the molecules in circular fingerprints, for example. It's a very simple way to describe molecules. You just have well, you can represent your molecule as a graph in this case. So you look at a heavy atom, the atoms in the next layer and the next layer. It's very quick to calculate. It works actually pretty well. You can also use learned representations, of course, today, but in practice that actually works nearly as well. And then you can build a random forest or even a naive Bayes model if you want, in order to predict those class memberships. And so basically in a situation, in a new case, you can put in a new molecule, you can predict the class. So that means a class membership, in this case is a Bayes classifier. So that's the log likelihood here. So it's basically this molecule is 10 to the 95 times as likely to belong to the class than not to belong to the class. Of course, it's a vast overestimate, but it basically means that this target is the one where the algorithm thinks that molecule is most likely to bind to, or that class membership likelihood is highest. And then you can actually say, you can predict targets for molecules. That is, in practice, that is very relevant. If you have a compound, you see an effect in a living system, you don't know why you see that effect, you can actually use your data that you have already, and from a phenotypic screen, as it's called in pharma, you can predict what the reason for that effect is. So in this particular case, the machine learning models work quite well. What is characteristic? We have lots of data and we have quite homogeneous data, right? And in those cases, in early stage drug discovery, we actually can use machine learning models. Yeah, there's a few disadvantages, of course, as well. It covers only known chemical space. There's lots of data biases in the model, and that's quite a problem. It's difficult to disentangle biases and information anyway, in particular in this case. Yeah. But in any case, if you have lots of data, you have homogeneous data, you can use models in early stage drug discovery. But that's actually not the case in many, I'll skip those slides, but that's actually not the case in many situations that matter in vivo. Yeah. Okay, so what can we conclude about this flat earth view on data? In cases where we have lots of data and homogeneous data, we can predict models. They can be helpful as a hypothesis generator. But in practice, our data we have is much more conditional. So what does that mean? Let's go back to the slide I was showing before, and why that is actually very naive. And this is kind of the take-home message in case you work on drug discovery projects at any stage in the future. The labels that we were assigning, they are by and large quite meaningless because they depend on the conditions you measure at. So for example, we have a link here, a particular compound is toxic. That depends on the dose, right? So if you dose a compound at high enough a concentration, nearly any compound can be toxic. Or a particular compound binds to a target. Well, it depends on whether the compound reaches the target tissue. Let's assume we work on a brain tumor and the compound gets or doesn't get into the brain, right? Then the prediction whether the compound binds to the target really depends on the distribution of the compound in the body and whether the compound actually reaches the target tissue. Or a particular compound treats a disease. Well, diseases are not homogeneous concepts. So if you work on knowledge graphs, for example, and you use some disease ontologies, diseases are not discrete entities. They're actually symptom clusters. Yeah? If you have a disease, you actually can have a genetic cause for that, or you can describe it on the symptomatic level. Yeah? And these are very different levels. If two people of you have the same mutation that in principle should lead to particular disease, it might be visible in one case, but not in the other, right? Depends then on nutrition, on some other mutations that you might have. Yeah? So sometimes basically you have different layers of biology and you don't have a unique label you can assign to a particular data point. Yeah? And that's a huge problem when it comes to in vivo data. Yeah. If, for example, water can be toxic, right? So imagine sometimes you have computer science papers that try to classify whether compounds are toxic or non-toxic. Yeah? And then they're very happy that there's a whatever 99% correct classification rate. But as you can see here, that's one report. If you drink lots of water, 5 liters in a few hours, yeah, you change your electrolyte levels in the body, right? And that means that approximately 5 liters is the border here. And so that actually leads to hyponatremia, as it's called. You have a lack of sodium in the blood, right? And that changes your heart currents, and then you die basically. Yeah? But water, of course, you wouldn't label as toxic if you build a classifier, right? But the question is then, how do you take that conditionality into account, right? You can take it into account as a parameter, but the problem is quite often you don't have enough data. Yeah? You don't measure every compound in every biological system at every concentration. Yeah? You don't have that data. Yeah? And so quite often basically you are operating nearly in a vacuum, yeah? And you don't know, you cannot label your data in any meaningful way. And that exists, unfortunately, in nearly every area of data that we have in drug discovery. Those is one of those problems, but also if you look at, I mean, some of those terms here are probably quite much more from the life science area, but to just illustrate, a compound doesn't only bind to a protein. Yeah? It can have dozens of different effects on that protein. Yeah? So if you have an annotation that just says compound is active on a protein, that's insufficient. Phenotyping, for example, as well. How do you describe a disease? Yeah, I already gave the example. You can describe it on the genotypic level, on the phenotypic level. Yeah, but quite often, if you go to the doctor, Quite often there are diseases that actually overlap in symptom clusters. If you think about depression or anxiety, for example, yeah, these are not two separate diseases. Yeah, they overlap in their symptoms. Yeah. And if you have data that is labeled with one or the other, yeah, and you put that into a machine learning model, that's actually basically you have two different annotations. You treat as distinct, but they overlap in the symptom clusters, yeah, which means that they actually partially the same thing. Yeah, and that is very difficult if you train models. Yeah, you don't have properly labeled data, basically. Yeah, and that problem exists across all of the different types of data that we have in drug discovery. Yeah? This one is from pharmacology, right? You have different types of activity. How do you phenotype? Then biology, we can characterize on very different levels. We can measure sequences, do genome-wide association studies, for example. Why do we do so many studies of this type? It's probably not because we should, but because we can. Okay, and that's always, if you do some science, always ask yourself the question, is it because it's the right thing to do or because it's an easy thing to do, yeah? And quite often there's more technology push than scientific pull, yeah? So there's lots of data generated because we can and not because it's the right data, yeah? And of course, an iterative process, right? You need to have some explorative study to see what can you see in the data. Then you need to have it set up in the next round, basically, to generate the right data for a given purpose. So computer science is tremendously powerful, but is our data in the field of drug discovery evolved? Quite possibly not. That is the point here. There's two different areas you can operate in, and that already became clear, I think, from one of the previous slides. So one is early discovery of proxy space or the in vitro space, and then there's the efficacy safety space or in vivo space. And so if you're in discovery or proxy space, you often have simple readouts. So you measure, you have an isolated protein assay, you measure, does a compound bind to that isolated protein, okay? It's a very simple setup, you can reproduce it very well. Unfortunately, that is not the situation you have in a living being, where you actually have a cell, for example, and the question is, does the compound cross actually that cell border in order to get into the cell? Yeah? But this is very simple data you can generate. Where you can generate large number of data points that are very homogeneous. So you have clear labels, you can train your supervised model, okay? On the other hand, you can have data from the in vivo space, where you have quantitative data, much more complex models, and your classes quite often depend, for example, on dose. It is really tricky to generate data on that. Animal data, for example, if you generate animal data and you have a particular clinical study, animals react differently if you have the cage on the top of the pile or the bottom, because light is different. Yeah? Animals react differently if you have a female or male handler. If there's a male handler, then the stress levels are different, okay? So, and that all these are confounding factors in your data. So if you do machine learning, yeah, and you have data from the life sciences, don't just take that as a matrix or whatever, or as a vector of data, yeah? And you need to understand the data, where it comes from, yeah? Especially in vivo data is very, very tricky, incredibly tricky. Yeah? And so if you have data from that area, in vivo data, it's quantitative. Things depend on dose, for example, right? You have much more complex models. If it's complex models, you have less data, and the data is much more conditional. Yeah? And that's then difficult from the machine learning angle, because you cannot just label data and say a compound is toxic or so. Yeah? It depends on dose, and if your cage was at the top or bottom of the pile, right? But then the question is, how do you put that into the model? And also, but the questions you ask from early stage models and late stage models are quite different. You can operate in a discovery setting. So for example, virtual screen, let's say, yeah? Virtual screen means you have many compounds to pick from, 10 to the 10 or so, and you say, give me the top 10,000 or so, I will test an experiment. That's a discovery setting, and that's easy for the model because there are many solutions in chemical space. You only need to pick some suitable solutions, right? Unfortunately, if you're at late stage in drug discovery, you need to make a prediction for the compound that is in advanced stages. Yeah, so you don't have the luxury of picking 10,000 out of a million or so. Yeah, you need to make a prediction for that particular data point where you might not have a suitable data, yeah, for the prediction. That's a much more difficult setting, and that, of course, is a problem because, as we've seen here, we have much less data and much more complex data in that area, okay? So we're asking for something more difficult from the model, yeah, although we have much less and much more complex data available. And that's a problem in practice, yeah? In vivo means does the compound do the right thing in the living being, yeah? But that is actually more complex data, we have less of it, yeah? And we have that huge conditionality in the data, yeah? And that's one of the main problems in using AI. in the drug discovery context. There's also, we characterized the different areas of information we can operate in when we compare drug discovery, for example, with the chess or Go or some of the other areas where algorithms are very successful, right? And one of the main differences is if you play chess, for example, your system is completely defined by the setup of the different, the queen and whatever, the pawn and so on the chessboard, right? It's completely defined by that. It doesn't matter which day of the year it is, the temperature doesn't matter, and the name of the player doesn't matter, right? It's completely defined by that setup, yeah? And that is different from biology. You have many variables that actually, it's difficult to describe your system completely, right? The setup of your system. And that is very different in biology from some other areas, such as chess and Go and so on. Yeah, where actually the temperature might matter, yeah, and you cannot capture what is the expression level of all genes, where they're located in the cell and so on. You cannot describe the state of your system properly. Yeah, that's also in the Drug Discovery Today article discussed in more detail. An example of if you work with knowledge graphs, for example, in drug discovery, right? You might be tempted to say, okay, we have compounds that bind to targets, and those targets are in pathways, and those pathways have something to do with the disease. We've done that before as well in the companies we started. AstraZeneca does it as well. Any pharma company does it. But I want to point out why that is actually not as trivial as it might seem. So this is a compound, it's ketamine, that's been used as a street drug anesthetic for 50 years or so. So it's quite a well-known compound, which isn't true for all the compounds we know, but this is quite a well-known compound. But even for that compound, we actually discover more and more uses. So it was used first as an anesthetic, right? As well as a street drug, but actually it also has been discovered to be an antidepressant, but only 30 years later. It's also a bronchodilator, so you can use it against asthma. But if it's used as an anesthetic, you give it at 4 times the dose. Yeah, so that is that conditionality in the data, yeah, that I mentioned. So what does it tell us? Some of the annotations only get discovered over time. Yeah, so there are many missing. That's what it means, and they are very conditional based on dosing, for example. Quite often you don't know how drugs work. This is actually true for most of the drugs on the market. So there's a discussion of different receptors and so on here, of how the compound might work. And that means if you put that into a knowledge graph, firstly, many of the links are not known, right? So it's very incomplete, but that's not such a big problem. But also there's lots of uncertainty how the compound acts. Yeah. So imagine you have actually many of those links in your knowledge graph. You want to do some reasoning over the knowledge graph, right? You would get many false positives. And that's a huge problem. If you have PubMed or so, or you do NLP, yeah, one of the problems is that you link everything to everything. Yeah? And that means prioritizing, if you have a knowledge graph in drug discovery, prioritizing over heterogeneous data domains is very, very difficult. Usually you need 2 steps. The first one is more done by algorithms, and then you need an expert in the domain, pharmacologist or so, who goes through those lists and looks at those lists by hand. Yeah? But the information we have is very incomplete, right? So if you reason across that information and you multiply that by 1 million or so, yeah, with many more data points, yeah, then you actually come out with, lots of vague associations, yeah. It's very difficult to do that quantitatively in the end, nearly impossible. Yeah, sometimes you have also machine learning models that try to predict, for example, toxicity. We had already this one. Here's adverse reactions, and that's an area that's the most difficult data I've dealt with, I think, in my life. Let's see what comes next. But here, because adverse reactions depend on at least a dozen or so variables. Depends on the genotype of the patient. Then what is actually the disease endotype? So that means if you have patients that have a particular disease, they're more prone to showing particular adverse reactions even. Yeah? Then you can have a different dose. We had that already. And then if you have adverse reactions, not everyone shows the same adverse reactions, right? You might remember those leaflets that you have, yeah? And they tell you in 5% of patients or so, you see a rash on your skin or so, yeah? But who knows who those 5% are, yeah? But so you only see that in a fraction of cases. Quite often there's a gender, there's a sex bias, so you see that more in males or females. And sometimes you only see that after months of dosing. Yeah? So you dose and maybe co-administration with another drug. If you are older, 80 or so, you're quite likely to take 7 or 8 drugs. And then the question is, what's the reason for an adverse reaction? Yeah? Maybe it's the co-medication, and those interactions are very difficult to disentangle. Yeah? And so basically, does a certain drug cause an adverse drug reaction? Yes or no? You might be tempted to build a binary classifier, okay, but in reality, it depends on all those factors. Yeah. So I mean, so does a drug cause that adverse event? Yeah. very difficult to say, right? You have at least those 10 variables or so that may be confounding factors. And the data, and this is intrinsic to the data, right? And then you also have reporting biases, yeah? So that's even independent from that. So some events are more reported than others. The severe events are more often reported than, for example, headache. Yeah? Then you have a cultural bias. So people from some cultures, they actually report adverse events more than from others. Yeah? The nomenclature problem we discussed already, so terms overlap. Ontologies do exist, but some terms overlap, for example, and symptoms are quite often clusters. Yeah, so you don't have neat class labels, basically. Yeah, and so you have two sets of problems. One is more the biases from reporting, and the other one is intrinsic to the data that you have. Okay, and this is one slide that's trying to summarize why actually in the image domain, for example, CNNs in this case work better than neural networks in the drug discovery domain. So in the image domain, so you have an object here, you have a cat also, you can represent your image as a matrix, right? Then you can have your convolutional network, so basically edge detection or whatever you have in there. So you have AlexNet, let's say, also that operates directly on that image, yeah. And then you have a largely unconditional label, right? So object can be well represented as a matrix. The CNN operates on that matrix, and then you have the object label that's by and large unconditional. Of course, objects can be hidden and so on, but by and large that is still unconditional. In the chemical domain, that's actually quite different. So you have a molecule. That molecule can be in different conformations, different protonation states, yeah. So different, well, it can exist in different forms basically. It might react, so it might change basically. And then the question is, how do you represent that molecule? And there's tons of ways to describe that. And there are learned representations as well. And you can do that if you have enough data. Yeah? But on the previous slides, you've probably seen that lots of the data we have is conditional, right? So if we cannot even label and assign classes, toxic, non-toxic also, to the data we have, right? That is actually very difficult. We have only very small data sets that's conditionally labeled, right? And then to use those learned features, it's actually tricky, impossible in some cases. So we have an object that's not clearly defined, the representation. It cannot be learned in many cases because we have conditional data. Then the question is, how do we model that data? There's nothing as obvious to use as the CNN on the matrix representation of the image. And we have largely conditional labels, right? So that means there's actually much more tricky to learn from data in the drug discovery domain, in the biological domain, then that's even worse. The predictivity of variables is quite limited. So quite often, if you build a model for something in the biological domain, because people react so differently, bodies are tremendously different. Yeah? If the same thing happens in one body, it might have one effect. In another person, it might actually not have that effect at all. So what you see here in this particular case is positive predictive value and the hit rate of drugs that cause an adverse event. So basically what that means is we have, for example, in those cases here, we have a certain fraction of drugs, or let's take that data point here, we have a certain fraction of drugs that hits a particular target, so it's active against a particular target, and then causes a particular side effect, arrhythmia, for example. Yeah, but if a drug is active on the target, I see that effect, that side effect, only in 38 or whatever % of the cases. Yeah, and also if I see the adverse event, the number of compounds that are active on the target, so the other way around, is only about the same number, or 25% or so. So what does that mean? There can be many causes for a particular adverse event. And if I have a particular event that usually causes an adverse effect, that might be only the case. That might only lead to the particular adverse event in a minority of the cases. So basically, there's no strong causal links, yeah, when it comes to the particular observables in our system and the phenotypic outcome. And that's difficult if we construct, for example, knowledge graphs, yeah, and we have lots of those weak associations. Yeah, we don't know many of those associations that we have between compounds, targets, diseases. and many of them are conditional, right? And many of them are very weak, yeah. And that means it's difficult to just put information into a knowledge graph and then assume the knowledge graph will tell us something. I mean, the knowledge graph will tell us something, but how meaningful that is, that's the other question, right? So quite often it's difficult to quantitatively reason over knowledge graphs over information in our particular domain. And that question also exists, of course, when we generate data. So genome sequencing, right? That's genome England and so on. That's quite a big thing here. Why do we actually sequence? Because it's cheap. It costs about $100 or so to sequence someone right now, right? And that is one of the reasons where that data gets constructed. But of course, there's many other layers of biology as well. We can do genomics. We can measure gene expression. So how much of that particular gene do I have? We can measure protein levels. Proteins can be modified. That's called phosphoproteomics. So they can be active or inactive. Yeah? We can measure the metabolome, so other small molecules in the body that get metabolized and so on. And the question is, which type of signal for which disease do we actually find on which layer of that information, right? And that's not clear at all. And that's one of the problems right now, where you have lots of data that is generated in a non-hypothesis driven way. That's one of the learnings from a postdoc, I think. I worked there on non-hypothesis driven data as well. Wherever you can, work on something hypothesis driven, yeah. So don't generate a data pile and then look for the signal, yeah, but rather find out what you should generate and then analyze the data afterwards. Yeah, and this is I think the concluding slide for today. So this is the summary of the Drug Discovery Today article. There's quite a lot of focus right now on methods, computational methods or end-to-end learning, also on representations. But in many cases, it's actually important to ask the question, first to understand what's actually the question we want to ask, and then what is the right data to answer that question, right? And only then the question comes, what is the representation and what is the method to learn from that data? Because if you have a method, yeah, that is based on unsuitable representation, that won't work. And if you don't have the right data that actually matches the question or hypothesis you ask, that also won't work. So it's really important to read that pyramid from the bottom, yeah, to understand the question and ask the right question first, yeah, have then the right data for that, represent it in the right way, and then have a good machine learning method that is able to learn all the way down to answer that original question again. Okay. Yeah. I think here I would like to conclude. Are there any questions? Yeah, thanks. Sure. Yeah, go ahead.
Yeah. So if we weren't limited by, I guess, our technology in terms of data collection and cost and everything like that, is there a type of data that could address some of these issues with, you know, the lack of clarity about what the label means, like having lots of conditionality? Like, is there one type of data which would be helpful for accelerating this kind of machine learning?
No, we don't even know that yet. So in some sense, we don't even know which direction to move into. It's more spotlights right now. I mean, there's a chance discovery that upregulation of that gene drives that cancer. Yeah. So you know one single association of in this area that type of data is marginally predictive. Yeah? And no, we don't even know that. It's so much technology driven. Yeah. Sequencing or gene expression, whatever we can measure. Yeah. So it's like a beam of light. Yeah. That gets put on that into that direction, yeah, and then people look for what do we actually see, which type of signal do we actually find in that area, yeah. We don't even know where to look for what in detail. Not really. Yeah, anyway, please.
How do you make targeted verification? Like, are there specific ways to collect data from people which are currently available?
Yeah. Targeted medication in cancer, for example. Yeah, cancer is okay as an example. Yeah. So it's very different from chemotherapy. Yeah, if you have a cancer and you do targeted medicine, that means you understand what drives the cancer. Yeah, there's HER2 or whatever. I mean, there's some certain things that can be upregulated, for example, and you target a particular process that is upregulated in that cancer, which means you target only the cancer. Chemotherapy kills all cells. Yeah, and because cancer cells divide more quickly, if you kill all cells, you kill more cancer than healthy cells. But of course, you kill too much overall, right? So that's not good. But targeted therapy basically targets the process that drives the cancer. So you need to understand stratified patient stratification is the key here. Yeah? You need to understand who are the patients that have that cancer and that genetically also have a certain process in place that stimulates that cancer growth. So you need to find the slice of patients that are homogeneous with respect to that biological process. Yeah? And then you can target that process. Yeah. So you can, for example, sequence all patients and you can say, do I get subgroups, yeah, mutations, for example, and if you get mutations that are very much enriched among your patients, yeah, that actually have that particular cancer, you can assume that's the causal reason. Then you can try to target that pathway, for example. Yeah. It's much cleaner than chemotherapy, yeah, much cleaner. Yeah, it works only in some cases right now, but still it's much cleaner than chemotherapy. Yeah. Any other questions, please?
If you think that the whole lot of data are big extended things, do you not think that many people with chemistry and machine learning understanding should be working with the science and use that
Okay, so, sorry, because of the noise, could you just repeat that?
So if you think that the limiting data are the biggest value,
Yeah.
do you not think the chemists with like chemistry and machine learning background should be pushed towards like chemistry
I know. I think it's not one or the other. In this chemical, you need both. Yeah? Chemical synthesis prediction can tell you how do you make the compound, but which compound should you make? Okay, that is the other question. Yeah, and for that you need to understand biology. Chemical synthesis prediction, there's lots of focus on that right now because it's relatively clean data. Yeah, and it's also useful relatively. I emphasize the word relatively. Yeah, but because it's labeled data, yeah, it's kind of trying to build a model way of data. But the question is, how do I make the compound, but which compound should I make? You need both. You really need both, yeah? Because otherwise you don't know in chemical space what you should focus on either. You really need both, yeah. Yeah. Because disease, you need to understand the biological drivers of disease. Yeah? That's the final application of your compound, basically. Yeah. Anything else? Any other question? Yeah, please.
What are some things that you're hopeful about? What are some things that you find the most exciting?
Yeah. For example, I mean, so one of the things we do with our company now, Terra Lumina, that's, for example, having dual starting points. Yeah, one is historical use from traditional medicines, for example. Yeah, so you have Ayurveda, traditional Chinese medicine. Also in Europe, you have virtually every country had traditional medicines, right? You always needed to treat sick people, and you have those annotations in knowledge graphs right now. Yeah. So you can combine that. So you have phenotypic evidence that compounds works, and you also have the Western target-based evidence. Yeah, and so you can come from both ways basically. Yeah, and to say, give me those areas where we have evidence the compound has been used and has been efficacious, and where I know how it works. Yeah, and if you have both pieces of information, that can be very useful. For example, yeah? That's where I personally believe I would focus on. Yeah. Because then you understand how something works, which is good to know as well for side effects and so on. Yeah, and you have real evidence from the clinic. Yeah, so you don't have the translational problem so much. Yeah, you have evidence about clinical efficacy already early on. Yeah, that's the point. Yeah, you can see it in this way. You can see it in this way. Repurposing usually in the current legal terminology is more if you have a compound approved on the market and use it for a different purpose, yeah, that is repurposing across time, if you wish, right? Because that's the historical use to repurpose it for something today. Yeah, in this sense, it's repurposing. Yeah. Yeah. There was one. Yes, yeah.
There's a place for the evolution of regulation where we've got this kind of process of trials where we're collecting these kind of details around responses over long-term infections. Because at the moment, obviously, the structure of the research funding is and silos, companies, how are we ever going to get this data on the responses of patients over the long term? That's the bit that's missing. We don't have enough of that. We do know we need, we don't have access to kind of broad data sets. So the only way we're going to get that is if surely it comes in the regulation, like the to bring drugs to market, you also have to have this kind of monitoring over a longer term, not just adverse events, but actually monitoring.
I mean, there's three sources of information for that. One is companies. Some countries, and the UK is doing quite well. Scandinavian countries are even better, or Estonia, and they have longitudinal patient records. Yeah? So they have that on the national level, and that's fantastic. In Finland, for example, is always one of the prime examples. Yeah? The UK is not bad either, I have to say, because of the NHS. But those longitudinal patient records, you have that for really big cohorts, in particular in Scandinavia. Yeah, and you can retrospectively, you can analyze. Does a person who had, I don't know, a particular disease in the 30s, is that more predisposed to have a particular disease in the 60s? Which drugs did they take in between, even 10 years ago? In some Scandinavian countries, you have those records. That is crucial. That is crucial, because otherwise, only then you can discover something interesting in the data. Otherwise, it's only a spotlight. You only analyze the association between two data points. In those types of records, you can be longitudinal across many patients, and across any disease that was annotated, basically. Now, the patient registries in the Nordic countries, that's the best example. And usually, if you write an EU grant, for example, or other grant, you can have access to those patient registries as well. You need a collaborator in the country or something, maybe, but you can access those as well. And in the UK, I think via the NHS should work as well. But otherwise, the Scandinavian countries are very good in that. Any other questions? Okay, great. Thanks a lot then, everyone. Have a good evening.